So. All right. Thanks everyone for coming. This is a very special day. Welcome to Aria's Defense. So we are joined by her examination committee, Jill Bargalotti from the Hunter College. She is on Zoom. Linda Spatz from the CUNY School of Medicine. She's in the audience. And then we have two external advisors, Dr. Alan Bazar from the University of Bordeaux in France, and Dr. Larry Zweifel from the University of Washington in Seattle. And at this moment, I would really like to thank the advisory committee. As everyone knows, the external advisors have only to be present for the second level examination and for the defense, but everyone made it to all of the committee meetings and was really, really helpful for Aria and myself to guide Aria. My name is Andreas Kottmann. I'm the advisor of Aria. So Aria, for today, I'll just go quickly through the procedure of the day. So there will be some introductions. Then and I will give a short introduction to the dissertation project. Then Aria will give a pre-defense seminar, which is followed by a question and answer session. And after that, we ask everyone to leave the room with the exception of the advisory committee, which would be just me and Linda. Everyone else will be on Zoom. And there will be a celebration around 2-ish on the third floor. Everyone is invited. So while I do not know the results of the examination that happens after the seminar, I think it's worth sticking around. There will be ample reason to celebrate. And the examination sounds so harsh, but it will be more a discussion. And I'm really looking forward to this because this will be one of the few last chances I have to discuss science with Aria in person. So with that, let me get into the introduction to Aria. And so I have to remind you of COVID-19 in many different ways in order to understand her project. And it was a horrible time. And that is when Aria joined and came from New York to Seattle. And you have seen all of these graphs. So we were just in the peak of the first wave. Aria came to New York. And what was already impressive is that from the rather safe West Coast, she came to New York and she didn't immediately turn around and went back home. She stuck it out here. And so she joined the lab at the tail end of this first wave of COVID. And then we closed our lab. We just maintained the animals, and then we opened again. And Aria asked whether she could join the lab. At the time was really horrible, right? This is sort of a typical picture, and the city was deserted. And what kept us going was, well, let me first say this. So Aria came from the University of Washington, and like for many other graduate students, she took peer mentoring and mentoring of younger scientists very, very seriously. And she started already at the University of Washington, where she taught calculus and science communication in an outreach program for minorities. Here at CUNY and at the Sophie Davis School of Biomedical Sciences, she taught and mentored 3 Rudin Fellows, Lillian Lee, Michael Holmes, and Leila Morgan. She also, more recently, was very much involved in getting Mia Singh and Nicholas Forgione going to Sophie Davis undergraduates. She was also involved in mentoring to master students in the cognitive neuroscience sub-program at the VC. She's an organizer of Brainy and Uni Science Com. She was highlighted in the the spotlight. She also taught in human genetics throughout her time here at the CUNY School of Medicine, and her student evaluations were always excellent. Okay, so with that, let's switch back and then let me color a little bit more the situation in 2020 when we started. So while we were all closed, I, like many other of my colleagues, joined a forum organized by Columbia University that met once a week. It was organized by Andrea Colicano. And he managed to draw in hundreds of scientists from not just the local area, but from the whole nation and internationally. And it really opened up my understanding of what was going on. It was an immense service to the science community. And what happened already a few months after the first peak of COVID infection had passed, it became clear that some patients suffered from long-term neuronal complications after they had cleared away the virus. And the term long COVID was coined for the first time. And it became clear in digging deep into the literature that there was old data that suggested that the receptor for the virus is indeed expressed in the brain. And we discussed this, and among many others, Dave Sulzer, who was sort of a preeminent scientist in basal ganglia and area in the brain, we'll talk more about in just a second, and is sort of our subject. He said, well, it might not be worth diving into this because the data really is flimsy. Well, the data is flimsy, and I'm not convinced. And before someone really shows that the receptor called ACE2 has a physiological function in the basal ganglia, it's probably not worth investing resources into this possibility. And so we discussed this, Aria and I, and Aria and I thought, well, if this is so crucial, and we proposed to be interested in the basal ganglia, and this is where maybe the receptor of the virus is, maybe this is something that we should find out, and maybe that could contribute to solving some of the issues with COVID-19. So this became an independent project, which became much deeper than this sort of first idea of trying to find out whether there's a direct link between COVID and the virus infection and dysfunction in the brain. But what Aria will tell you later on, that there is a function of that receptor in the basal ganglia. So what are the basal ganglia good for? And I would like to guide you through this so that you understand the context of the work a little bit more. So the brain has two ends, basically. There's a cortex up there. So this really on one side, there's the cortex, on the other side are motor neurons in the spinal cord. And the only way anything can come out is that the cortex elaborates. It's through the common pathway which connects motor neurons with muscles. And so, as Sherrington said 100 years ago, to move things is all mankind can do, whether in whispering a syllable or in felling a forest. The cortex is concerned with ideation and planning. And what the basal ganglia in between do, the heterogeneous population of circuits and nuclei underneath the cortex, is to translate that thought of the cortex into action. Or as others said later, the cortex broadcasts the wish to the rest of the brain, but the basal ganglia may ground that wish. And so how I picture what the basal ganglia are is an advisory board. The advisory board checks on the ideas of the cortex and advises back. And what happens in the basal ganglia is that the plan is vetted. It also builds institutional knowledge. What worked before would work likely in the future again. It builds internal representations of the world and keeps tabs on the validation of these representations, on the validity of these representations. So what it does, in an essence, it constantly updates, and that leads to learning. So the moderator of that advisory board that gives feedback to all the advisors sitting around the table is dopamine. And so if the advisory board becomes corrupted, the consequences are severe. The basal ganglia are involved in a litany of diseases: schizophrenia, Tourette's, addiction, impulse control, Parkinson's disease. And as we heard earlier, there was a discussion that some of the long COVID pathology might also relate to the basal ganglia. So the basal ganglia is a hotbed of pathology. But despite extreme effort over 2 generations of molecular neurobiology, no disease-modifying therapies are available to any of these diseases. And with that, Aria.
Okay. Thank you all for coming. I really appreciate it. Let me share what I've done the last 5.5 years with you. So as my title says, I'm looking at how the local ACE2 enzymatic activity can modulate cholinergic interneuron function within the dorsolateral striatum, but that's a mouthful. So I usually just say I'm looking at the role of the renin-angiotensin system in the basic ganglia. So we're at a medical school here, and when I talk to people about the renin-angiotensin system, most people know of it in its relationship to regulating blood pressure. And in large part, this is because this is how it was first identified. It was identified long ago in the late 1800s, and it was seen that there are these enzymes in these products that are produced in the kidneys and liver, and they seem to be able to have large effects on controlling blood pressure. And for a long time, this is the main thing it was known for. But it wasn't for an additional 70 years before they found that there was a separate renin-angiotensin system that was also present in the brain. And this was very exciting because many of the peptides themselves cannot pass the blood-brain barrier. And so they're being locally produced within the brain, and that suggests that they must be doing something, because nature is cheap, as they say, and if it's not using it, it's going to get rid of it. And so coincidentally as well, this same work that first showed that it was in the brain was also looking at its role within the basic ganglia, finding that there is a large amount that persists into the adulthood. But at this point, there was not a lot understood of what is RAS, or the renin-angiotensin system, doing in this area. So when I first joined the lab, we were very interested in this as its relationship to COVID, but this is more than anything a Parkinson's lab. And we were interested in the fact that there's mounting evidence that suggests that the renin-angiotensin system plays a role in Parkinson's disease vulnerability. So this is a lot, but this is more or less a lot of the papers that influenced me early on when I started approaching my project. But specifically what I found was interesting is these retrospective case studies looking at Parkinson's disease patients who were also being treated for high blood pressure. And what they found is that specifically when you use RAS-targeted drugs to control blood pressure, you have a reduced occurrence of one of the most common side effects of the gold standard L-dopa treatment, which is L-dopa-induced dyskinesias. So these patients taking this specific class of drug seem to not have as bad of a side effect with dyskinesias. And further work also showed that it's not all RAS targeted, but specifically RAS receptor blockers that have the strongest effect on reducing this pathology. And what's good is we can use these same drugs and look at what it's doing in a mouse model of Parkinson's. And they found that in an AAV alpha-synuclein model of Parkinson's, where you can model for early progression of Parkinson's, when you co-treat or when you treat these mice with these RAS receptor blockers, there is a slower progression of the disease early on. And so this was very exciting, as this might be some mechanism involved in reducing the vulnerability of Parkinson's patients. So the pathway is very long, but I'm only mainly interested in a short part of it, so I'll introduce it to you. It starts with the angiotensinogen peptide, which is released in the periphery, mainly from the liver. But in the brain, it seems to be mainly produced by neurons and glia. And it's first cleaved into angiotensin 1 by renin. So that's the renin and the renin-angiotensin. And then it's cleaved again by angiotensin converting enzyme 1, or ACE, into the first active signaling peptide, angiotensin 2. And what's interesting about this peptide is that it can have two opposing effects depending on the receptor that it interacts with. So first, it can bind to angiotensin 2 type 1 receptor, or AT1R. And in the periphery, this has the classic blood pressure raising effect through vasoconstriction. But there's evidence that shows that in the central nervous system specifically, it has an oxidative inflammatory response. And this can exacerbate Parkinson's disease as well as other neurodegenerative diseases. But the same peptide, angiotensin 2, can also bind to angiotensin 2 type 2 receptor. And what's interesting is this has all of the opposing effects through downstream signaling. So in the periphery, it can balance blood pressure by having a vasodilation effect. But then in the brain, it is antioxidative and anti-inflammatory. And so this section is the most well-studied of the renin-angiotensin system. It's the most highly expressed in the periphery, and it's called the canonical pathway. So most of what we know is about the canonical pathway. But the pathway doesn't just stop here. And this is where my main character comes in. So we can further cleave angiotensin 2 into angiotensin 1,7 by the ACE2 enzyme. And so this is also the target of COVID-19. This is how you become infected. And so once we make angiotensin 1,7, for the first time, we have a peptide that can entirely oppose these oxidative inflammatory steps. And it's not just through downstream signaling. What's unique about MasR is that it can directly inhibit AT1R as well as having these downstream opposing effects. And so through the balance between these two, you have a way of counterbalancing this oxidative balance, I guess I'll say. So this side of the pathway is less well studied. It's considered the non-canonical pathway, and it's more recently that work has started looking into this. But what's interesting about it is that this division between canonical and non-canonical also overlaps with what we know about the renin-angiotensin system and Parkinson's disease. So when you have shifts towards the canonical side, either through targeting ACE, angiotensin 2, and AT1R, all of these types of manipulations promote the progression of Parkinson's disease and increase the occurrence of levodopa-induced dyskinesias. But when you shift towards the non-canonical side by targeting ACE2, angiotensin 1,7, and MasR, now you are protected against Parkinson's disease, and there's a reduction of the occurrence of levodopa-induced dyskinesias. So I've been talking a lot about Parkinson's and the basal ganglia, but I'll give a little primer on it. So specifically, Parkinson's begins in the ventral midbrain, where you have loss of dopaminergic neurons in the ventral midbrain, specifically the substantia nigra. But this localized degeneration doesn't only affect this one area. Over the course of the disease, you have the early onset symptoms that are primarily targeting the nigrostriatal pathway or the substantia nigra into the striatum. And this is where you get the classic symptoms people talk about. So this is where you have difficulty with voluntary movement production. But as the disease progresses, you begin to have more like broader effects on the brain. So specifically, you can dopamine loss will begin to affect the mesocortical pathway, and this will give rise to symptoms relating to cognition, memory and learning, and motivation. And over time, it also spreads to the mesolimbic system, where you begin to have issues with emotion, perception, and reward. So specifically in our lab, we're mainly interested in the effects on the nigrostriatal pathway and largely focus on the dorsolateral striatum because that is the first area affected in early Parkinson's. So when looking into the literature, I'll use the word excited, but obviously it's a grim topic, but I was excited to see that there's evidence by a lab, it's my favorite lab in Spain, they do a lot of work relating to me, and they have shown previously that there is existence of the renin-angiotensin system within the nigrostriatal pathway. So starting on the substantia nigra side, they show that dopamine neurons express angiotensinogen, that precursor peptide, and renin, that first enzymatic step in the pathway. But then on the striatum side, medium spiny neurons, which are the most abundant neurons within the striatum, they also express some level of the renin-angiotensin system, specifically angiotensinogen, renin, that first enzyme, and then AT1R, which is the oxidative receptor. But based on the literature, there were still open-ended questions that I was interested in addressing. So specifically, are RAS components expressed within the dorsolateral striatum outside of just medium spiny neurons? This is a large area, and there's many types of neurons that play a crucial role in the function, including interneurons. And then beyond that, the previous work had mainly focused on the canonical renin-angiotensin system. But in order to have a balance in the signaling, I wanted to determine, is there a non-canonical RAS pathway within this area that can counterbalance that oxidation? So to begin with, I looked at ACE2. So this would be the enzyme, which can push us from that canonical to the non-canonical side. And what I found through immunohistochemistry in wild-type animals is that compared to a no primary control, there was a rather abundant expression of ACE2. And in co-staining for NuN, which can show us expression of ACE2 on neurons, we found that there was expression of ACE2 both on neurons within the dorsolateral striatum, but also on NuN negative cells. And so this would suggest, and it backs what people have seen elsewhere, that glia are also expressing a large amount of ACE2. And so this supports that there is the potential for a shift to the non-canonical pathway. And to further investigate that, I looked at the expression of the different receptors. So as I said, there's the AT1R, this is oxidative, AT2R, still canonical, but anti-oxidative, and then MasR, which is our main counterbalance. And when staining in the same area, I compared against my no primary control to determine, is it above a negative level? And what I found was that both AT1R and MasR seem to be expressed within this area on neurons. But what was interesting is that AT2R was not readily expressed. I had a very difficult time finding it. And that was an interesting finding because it means that between the canonical pathways, when there is angiotensin 2 expressed, it can only bind to AT1R. And I knew that this wasn't, this lack of expression was not due to inadequacies in the antibody, because we were able to use the same antibody and stain dopaminergic neurons in the substantia nigra and identify expression there, which had been previously seen by that the lab that had done a lot of the foundational work. And so this suggests that the main RAS receptors at play are AT1R and MasR. And so based on these stainings alone, we found it interesting that there seem to be these large cell bodies that are particularly high expressors of both AT1R and MasR. And to identify what we were, we co-stained with CHAT, which identifies cholinergic interneurons within the area. And we found that these large cells are actually cholinergic interneurons. And when compared against CHAT negative or non-cholinergic neurons within the region, we found that AT1R and MasR are particularly enriched on these cell types, not just in the mean intensity or the amount that they're expressing, but also when we look at whether or not they're positive expressors, which was based on expression higher than my no primary, we found that 100% of cholinergic neurons co-express AT1R and MasR, as opposed to 93% of non-cholinergic express AT1R, and then only 84% express MasR. And so this was the first aim of my project, and what it told me about the system is that ACE2 seems to be broadly expressed within this region, meaning that we can not only get COVID in the basal ganglia, but we can also push that oxidative pathway to the non-canonical anti-inflammatory side. And so what that meant is that when the Yes, when angiotensin 2 is present, it can be readily converted to the non-canonical side. We also found that AT1R and MasR were detected across the neurons in the region, but were particularly high expressed on cholinergic interneurons, where AT2R was not expressed, meaning that when angiotensin 2 is present, it can only bind to this inflammatory side. And CINs seem to be particularly sensitive to this ACE2 regulated balance because they express both sides, where MasR is able to directly inhibit this oxidation. Okay, so we were particularly excited about this because in the lab we love cholinergic interneurons, and it's not just because they're big and pretty, but they also have a key signaling role within the striatum. So anyone who knows about them, they don't make up that much of the population. So as I said, medium spiny neurons are about 96%, and then only about 1% of the cells in the striatum are cholinergic. But what they lack in population, they make up for in these beautiful large arbors through which they're able to exert a disproportionate role in signaling. And through these large arbors, they're able to integrate signals from many different other neurons within both the striatum and outside of the region. And in order to integrate these signals, they have to have a way of receiving them. And so what's interesting is they're positive expressors for many different receptors that are receiving information from these different sources. And what I've been able to add to the literature is that they also express AT1R and MasR. But beyond that, with ACE2, ACE2 cleaves many different peptides within the striatum, and many of those receptors are present on cholinergic interneurons. So ACE2 is able to manipulate the signals coming in to cholinergic interneurons. And so the reason we're particularly interested in CINs, as I said, is they play a large role in striatal function. They're implicated in some of the key behaviors, including flexibility and strategy shifting during learning, reward prediction error, and then also locomotor interruption. And some of the key ways that they're able to execute these functions is their unique interplay with dopaminergic neurons. So dopaminergic neurons from the ventral midbrain innervate into the striatum, and there's this unique balance between their signaling, where both cholinergic interneurons and dopamine neurons fire tonically to establish a base level signal, but changes in the rate of these signals has large impact on what's happening in the area. And so it's able to encode information about the salience or the importance of events, and as well as behavioral state information. And cholinergic interneurons and dopaminergic neurons can modulate each other's activity. So CINs predominantly communicate with acetylcholine, and they are able to control dopaminergic neurons through nicotinic acetylcholine receptors. And so these are predominantly located on the dopamine terminals and can trigger dopamine release from these axons even when there's not a cell body initiated signal. And then on the other side, dopaminergic neurons communicate predominantly with dopamine, and they are able to modulate CIN activity in part through excitatory D1, D5, but predominantly through D2 inhibitory receptors. And from this signal, they're able to produce a multiphasic effect in cholinergic interneurons. And so what we see is when there is an important learning event where you get this big dopamine burst, when the event occurs, there is an initial excitation in part from the excitatory and also from other inputs from the cortex. And then as dopamine increases, there's this big pause in acetylcholine activity that's followed by a rebound as dopamine returns to normal. And so in particular, this multiphasic effect has an important role in an animal being able to learn an important event. So based on all of this, I asked myself, how does ACE2 enzymatic activity affect cholinergic interneuron function, specifically the acetylcholine dopamine signaling dynamic within the dorsolateral striatum? So to do this, I used photophotometry, where I measured the amount of acetylcholine and dopamine being released using grab sensors. And what this means is that it's based on a GPCR with a circularly permutated GFP, where when either acetylcholine is released and it binds to this receptor, now we have fluorescence. And within the region, when I have a optic fiber implanted, I have to use this, when there are dopamine releases or acetylcholine, you get these flashes of fluorescence that correlate with the amount of acetylcholine and dopamine that's been released. And so in combination with this, we did something a little more sophisticated, and we paired it with an optofluid cannula. And so what this entails is that we have the classic optic fiber through which we can measure the fluorescence, but alongside is a local fluid injection port. And what this means is that I can inject pharmacology bypassing the blood-brain barrier, but also making sure that these initial effects from the injection I see are from the local changes and not from secondary effects elsewhere. And then, as you see, our paradigm includes a running wheel. So we have the mice head fixed atop the running wheel. And then over the course of 45 minutes to an hour recording, they're freely able to ambulate atop the wheel. So this was how we're going to measure acetylcholine dopamine, but I also need to be able to manipulate ACE2 enzymatic activity. And so based on what I knew about the system, here is the normal conditions where we have ACE2 expressed both on neurons and on glia. And we're going to approach this with two different techniques. We're going to use a genetic ablation where through that local fluid injection port, we will inject AAV-CRE that is on a HSIN promoter. And then after the AAV can ablate ACE2, what we end up generating is a chronic neuron-specific loss of ACE2 enzymatic activity. And this mouse was initially developed, in part, sold through COVID. It was expensive, but we're able to generate a unilateral genetic ablation, where ACE2 is still present, but it just can no longer cleave the peptides. On the other hand, we can also acutely inhibit ACE2 activity by locally injecting the small molecule inhibitor MLN4760. And so this, again, will be through that local fluid injection port. And as opposed to the neuron-specific, this injection will acutely ablate all local ACE2 enzymatic activity, including on glia. And so with this paradigm and the implant in the dorsolateral striatum, we are able to get traces that look something like this. So over the course of the trial, the mouse is running, doing stuff, thinking about things, and we can see these fluctuations simultaneously in acetylcholine and dopamine. And we were inspired by Guppy, which was this paper, and we wanted to take an unbiased look at dopamine and acetylcholine burst events. So what we do is we run a threshold over our moving mean raw trace, and this is a 3 median absolute deviation line. So all events that are above this are 3 median absolute deviations above, or they're significantly larger than this moving average. And so from our traces, we can isolate these important events and pull both the guide trace, which is what the trace was run on, and then also look at what's happening in the corresponding. So we can see when, in this case, when acetylcholine is high, what is dopamine doing at the same time? And from this, we're able to average and see over the course of this session under these conditions, what is the dynamic like between acetylcholine and dopamine? So based on my control animals, and this is about 9, this is more or less what the dynamic is for acetylcholine burst event. So when acetylcholine burst fires or asynchronous activity, it's preceded by a dip in dopamine and followed by a small rebound. And this overlaps with what we know about the relationship between acetylcholine and dopamine within this area, and that acetylcholine stimulates dopamine, but then when dopamine is released, it can inhibit CINs and they no longer fire acetylcholine. So based on this, we can begin to look at what is ACE2 doing with this dynamic. And so comparing our genetic ablation versus our pharmacological manipulation, when we just look at acetylcholine burst firing events, there's no significant changes under either condition. Neither in the amplitude with which they're occurring or the frequency of these burst firing events. But what becomes interesting is when we look at what dopamine is doing when acetylcholine is burst firing. So comparing the two conditions and looking first at this initial dopamine dip that precedes, there is a significant reduction in the amplitude of this event occurring before the dopamine burst or the acetylcholine burst. And it's approaching significance in the pharmacological manipulation as well. But what's interesting is that even though this preceding event is different, there's no significant change in either condition in the follow-up rebound of dopamine. And so that implies that there's some importance to that initial event. And to begin looking at that a little closer, we also looked at the correlation, the time correlation between these two different traces. And so what this entails is we run cross-correlation on the guide trace, which is acetylcholine, and then shifted our follow trace dopamine at the same time to see how they predict each other in time. And so what this means is that if I shift negative, so I move this forward, so what happened before is at the same time, we see that at a negative lag, there is a negative correlation between these two events. But when we shift positive, we see that there is a positive correlation between these two events. And this tells us about the relationship between them and how they can predict one another. So using this, we looked at the two conditions again, and looking first at that preceding dopamine dip and how it predicts the acetylcholine burst, we see that there's a significant reduction in this predictive relationship between the dopamine dip and the acetylcholine burst, but only in our genetic manipulation. And then similar to what we saw in the raw traces, there's no significant difference between this maximum correlation in the event that follows the acetylcholine burst. So now we can also do the same thing I've done before, but now we're going to look at what happens when dopamine burst fires. So when we look at the controls, we get a dynamic that looks something like this. And what's exciting is this overlaps with what we know about behavior using other techniques. So this is through electrophysiology, where when you have this dopamine burst firing event, you have the initial excitation, the pause, and then a little bit of a rebound. And so we wanted to see how does ACE2 enzymatic activity impact this dynamic. So once again, we're going to start by looking at the dopamine burst events first. And similar to what we saw is that there's not a lot of difference between these conditions. It did not affect the amplitude of these events, nor the frequency at which they're occurring. But once again, things get a little more interesting when we look at what the follow trace is doing at the same time as this first event. So when we first look at that initial excitation that comes before the dopamine burst, there's no significant difference in either the genetic or the pharmacological. But for the first time in the phybrophotometry dataset, we have an agreement between these two conditions, where both through genetic and pharmacologic manipulation, we're able to reduce the amplitude of this acetylcholine dip that follows a dopamine burst. And this was particularly exciting for us because when you look at behavioral data, the acetylcholine dip that follows has a lot to do with the ability to learn from that important event. So now we can do again the cross correlation, looking at acetylcholine lag relative to the dopamine burst. And similar to the raw trace, when we look at the acetylcholine initial excitation, in the pharmacological manipulation, there's no significant difference. But what's interesting is that in the genetic manipulation, this acetylcholine burst that precedes becomes more predictive of the dopamine burst to follow, which was not what we expect. But then once again, we can look at what happens after the dopamine burst, and we see that there is a reduced predictive relationship between the dopamine burst and the acetylcholine dip, which overlaps with what we saw in the raw traces, but it's not quite statistically significant in the pharmacological manipulation. So based on this part of my work, I can conclude that loss of ACE2 enzymatic activity, either through genetic or pharmacological manipulations, does alter this relationship between acetylcholine and dopamine in the dorsolateral striatum. So when looking first at during acetylcholine burst firing events, it reduces this preceding dopamine dip while also making the predictive relationship between this dip and the following acetylcholine burst less correlated. And then on the other side, when we look at dopamine bursts, there is the reduced amplitude of this acetylcholine dip, as well as this reduced relationship between the burst and the dip that follows. And so, as I talked about already, we were particularly interested in this acetylcholine pause as it is a key signal for learning during behavior, where dopamine bursts are known to signal information about the reward, salience, and surprise of the event that occurred. And then this pause that follows, there's a synchronous pause in cholinergic activity in response to this dopamine, is important for the ability to learn from that signal. So the current thinking is that this pause in acetylcholine allows for a window of plasticity through which cells, neurons within the striatum are able to make changes based on the information that's being transmitted. And there's interesting work showing that within these behaviors, if there is a larger, more precise pause, it improves the ability to learn either from the motor or the learning reward that is occurring. And in thinking more about this pause, we were interested in that some of the work that we are seeing also overlaps with what is known about D2 receptors in the same area. So under a similar condition, this lab was able to show that pharmacological and genetic ablation of D2R on cholinergic interneurons can also reduce this acetylcholine dip after a dopamine burst. And similar to my work, the genetic ablation was also a stronger effect in that it reduced both the duration and the amplitude, where the pharmacological only reduced the duration. And so this overlap was interesting between these effects because AT1R and D2R are known to have a functional relationship within the striatum. And so specifically what they find is that there is this cross correlation between these two receptors, in that when you take hex cells that don't express either AT1R or D2 normally, when you co-express them together, they're able to have this functional relationship with each other, where at low concentrations, co-expressing AT1R further sensitizes D2R to the agonist quinferol. What's interesting is that at high concentrations of this agonist, co-expressing AT1R is blunting that same effect. And so this seems to imply that there's this relationship between the activity of D2R and AT1R. And so the next question I asked was, how does AT1R inhibition affect D2R agonism on cholinergic neurons in this area? And so to investigate this, I took 4 groups of mice and I injected them IP with either saline, which should have no effect on either AT1R or D2R. I injected losartan, which is FDA approved. There's people taking losartan, and this is a potent AT1R inhibitor. And so what this should do is this should inhibit AT1R specifically. And then when to manipulate D2R, I took the same drug that was used in the previous, and I injected quinferol, which should agonize D2R activity. And then to cross-manipulate, I'm going to co-inject losartan and quinferol together, which should simultaneously inhibit AT1R and activate D2R. So after these injections, I put the mice in the open field for 30 minutes to assess changes to locomotion, and then sacrifice and stain the brains for the CIN neural activity marker PRPS6. So this should tell me the recent activity level of the cholinergic neurons in the area. So beginning with locomotion, it seemed that there was no significant changes by inhibiting AT1R as compared to saline. So over the course of the trial, they behaved quite similarly to the control saline animals, where unsurprisingly quinferol had an immediate and strong effect on the animals, where they kind of get real tight and don't move anywhere. And then at the same time, co-injecting losartan and quinprol had no significant difference than quinprol alone. So this would imply that in overall motor output, AT1R inhibition doesn't have a strong effect. What was interesting is in looking at the sections of the animals, looking at PRPS6, losartan alone did not have a strong effect on its own, maybe with more animals, but with these numbers, there is not a significant difference between saline and losartan. And at this concentration of quinprol, there was not a significant difference between the saline control and the quinprol, even though there was a strong effect in locomotion. But when we co-inject losartan and quinprol, so when we first are able to inhibit AT1R and at the same time agonize D2R, now we have a significant reduction in CIN activity as compared both to quinprol alone and saline. And so this overlaps with AT1R having this ability to regulate D2R activity. So going back to that initial paper that I talked about, their final panel in their project was looking at whether or not AT1R and D2R form heterodimers within the dorsolateral striatum, or are they closely localizing and able to functionally manipulate because of close proximity. And so what they did is they did a proximity ligation assay and then looked at all cells within the striatum. And what they found was that most cells seem to be positive for closely localized AT1R and D2R potential heterodimers. But what they don't investigate is any type of cell type specificity. So building on this work to support what I see with the pharmacological manipulation, I did the same process, but now I'm going to also stain for NUN and chat to determine if cholinergic interneurons are also showing this positivity. So the main experimental condition is taking D2R LL mice, so this means there's no outright manipulation, and then including both the plus and the minus probes. And so under this, when they are within 40 nanometers of each other, it will make this big fluorescent puncta on those cells. For my biological control, I stained chat-free D2R LL mice, and so specifically on cholinergic interneurons, I should see a reduction in these puncta because they no longer express D2R, while other non-cholinergic neurons should be mostly unaffected. And then finally, as a technical control, I removed the plus probe so that there should be overall no formation of this circular puncta because only one probe is present. So looking in striatal sections of these animals and co-staining, I see that similar to what the previous work found, about 70% of cholinergic interneurons are expressing AT1R and D2R close proximity possible heterodimers. And based on the biological control, we see that the chat-free manipulation was able to dramatically reduce the amount of puncta on these specific cell types. And then as well, with the technical control, there is very few puncta present on these cells. And this supports that cholinergic interneurons are one of these populations that have these close proximity heterodimers between AT1R and D2R. And so overall, overlapping with what we know, that dopamine from dopaminergic axons are able to stimulate D2R and thereby inhibit CIN activity. And building off of what previously is known that AT1R and D2R are able to cross-manipulate each other. And my work shows that AT1R and D2R are closely co-localized on cholinergic interneurons, consistent with heterodimer formation, and that inhibiting AT1R increases this D2-related reduction in CIN activity, potentially through a loss of the ability of AT1R to inhibit D2R, allowing for a more dramatic reduction in CIN activity. And so through this work, I am proposing a model in which the renin-angiotensin system can modulate CIN activity through this AT1R-D2R crosstalk. So I've been talking this whole time about how important CIN activity is, but I think most lay people in particular are most interested in behavior. And so, what CINs are particularly well known for is their ability to regulate these striatal output pathways. And one of the main ways that we can assess this is through locomotion. So the balance between the two hemispheres of CIN activity is also important for the ability to make turns. So if you have balanced locomotion, you go straight. If you have a unilateral ablation, now you can induce turning, and CINs have been implicated in this. And then on top of that, CINs are also very important for the ability to learn and to change strategy as you begin to determine what the best course of action is. So CINs are very important for this. But what's exciting is that some previous work has indirectly of CINs suggested that RAS signaling within the striatum has similar effects on these behaviors. And so to investigate this, I first asked myself, does ACE2 ablation in the dorsolateral striatum alter locomotor activity, specifically in the open field? And so to do this, what I did is I took my fibrophotometry mice, which had the unilateral implant of the cannula, and so I manipulated ACE2 only on one side. And what we see is at week 0, when there is no genetic manipulation, there is no significant difference in the percentages of turns between these two conditions. If anything, there is a bit of a contralateral turn bias, but that's also because they have an implant on only one side. But within 3 weeks after injecting the CRE into the ablated animals, we see that there's a significant ipsilateral turn bias that arises that is not seen in the unmanipulated control. And this difference is maintained over the week that I followed this particular cohort. And when looking specifically at a group of cohorts at week 14, so well into the ablation, we see that this rotational bias is maintained, where there is a significant ipsilateral turning compared to the non-manipulated controls. And this turn could be a result of 2 different things, but we find that it's not because there is a decrease in contralateral turning. This difference arises because these mice begin to, there's an increase in ipsilateral turning or towards the direction of the manipulation. And so as these were a unilateral manipulation, I was wondering, is this something that we would see in bilateral? There would be overall increased motor learning. They're just going to move a lot more. And this didn't end up being true. So when looking both at just a unilateral ablation as well as a bilateral ablation, under these conditions, there are not significant changes to overall motor output. They're not just moving more. And so this suggests that there's some difference in action selection of turning selection as opposed to an increase in movement overall. So the last experiment that I will talk about is probably my favorite. Does ACE2 ablation in the dorsolateral striatum alter motor learning? And so to investigate this, we're going to bilaterally ablate ACE2 in my experimental animals. And then once the manipulation takes, we challenge them to an accelerating rotor rod paradigm. And what this entails is that the mice are placed atop a rod that begins to rotate over the course of 500 seconds, starting at 4 rotations per minute, accelerating to 40 by 300, and then maintain for 200 seconds. And so the task of the mouse is to stay atop the rotating rod, or to learn how to do that over the entire course of the trial. And they're not very good at it to begin with, but as learning progresses, there is an increase in the latency to fall at how long they're able to execute this task. And what was exciting is that when comparing our 2 manipulations, the mice that had ACE2 ablated on both sides were not able to learn on this task as well. So looking first at the mean latency to fall, or the average over the 10 trials over the course of the 7 days, they start out not as good as the controls, and they also plateau at a lower rate. And when you look at the maximum latency to fall, which can tell you their ability to perform the task at least once, they start out lower, but by the final day, there's no significant difference between the controls and the experimental ablation. And so these mice are able to do the task at least once, but they're not able to maintain this performance over the entire trial. And so this suggests that ACE2 ablation is having some type of inhibitory effect on their ability to learn. So overall, based on these final experiments, I can conclude that manipulating RAS through the ACE2 ablation has behavioral consequences in the dorsolateral striatum. So specifically looking at unilateral, it induces an ipsilateral turn bias, suggesting that ACE2 contributes to balanced action selection without changing overall locomotion or motor output. And when looking at the bilateral ACE2 ablation, there is an impaired performance on the accelerating rotor rod, suggesting that ACE2 normally is able to support this motor learning. And so based on these experiments I've talked about, the key findings that I would like to highlight are based on the dorsolateral striatum of the cells present. Most seem to be expressing ACE2, the ACE2 enzyme. And so this means that when angiotensin 2 is present, it can be shifted to the non-canonical anti-oxidative angiotensin 1,7. And CINs are particularly sensitive to this because they express both AT1R and MasR, which are the targets of these peptides. And as I said, MasR is a particularly good counterbalance because it can directly inhibit AT1R activity. And then through the middle part of the work, D2R seems to have a unique relationship with AT1R where they're able to balance each other's activity through a functional heterodimer formation. And so when you inhibit ACE2 activity, particularly through genetic ablation, you have these key findings of the reduced acetylcholine dip and the decoupling of acetylcholine dopamine, the ipsilateral turn bias under unilateral ablation, and then the hindered motor learning when you bilaterally ablate. And so my proposed mechanism of how all of this can fit together in a model is that when you inhibit ACE2 activity, either through genetic or pharmacological means, there is an imbalance in the amount of the angiotensin peptides, where you have reduced angiotensin 1,7 and increased angiotensin 2, which overactivates AT1R, reducing this counterbalance between ACE2 or AT1R and MasR. And this reduces that counterbalance ability of MasR to inhibit, which changes this relationship between AT1R and D2R, where D2R is too repressed, changing how CINs are able to interpret dopamine signals. There are, of course, future directions that would be very exciting to address. One of them, as I talked about before, is ACE2 is able to cleave many different peptides that are present in this phase of ganglia. And many of these are also expressed or CINs are receptive to them. And so as a proposed future direction would be based on my work looking at the genetic ablation of ACE2, where we can generate this decreased acetylcholine dip, ipsilateral turning, and hindered motor learning, we could investigate the role of specifically AT1R by looking into a genetic ablation of AT1R specifically on CINs, where in this case, based on the proposed model, we should have the opposite effect to what we saw with the ACE2 ablation. So instead, we should have an increase in the acetylcholine dip, potentially contralateral turning, and facilitated motor learning. And based on the model itself, even if we inhibit ACE2 within this genetic ablation, AT1R should still predominate these outputs. So even with ACE2 change, there should still be this increased acetylcholine dip, contralateral turning, and facilitated motor learning. And to end off on why everyone should care, there's obviously the clinical relevance of the project. So as I talked about at the beginning, L-DOPA is still the gold standard treatment for Parkinson's disease, but chronic use of L-DOPA also has severe side effects, in particular, the L-DOPA-induced dyskinesias, which are very debilitating. And what we know about LID is that CIN hyperactivity is implicated in this dyskinetic movement. And there's been preclinical work that shows that modulating this striatal CIN activity can in some ways reduce LID severity. And so my project in suggesting a role for the renin-angiotensin system on CIN specifically is an avenue through which we can understand and target LID in particular to either through ACE2 upregulation or through the FDA-approved angiotensin receptor blockage. And this might be a therapeutic approach to mitigate L-dopa-induced dyskinesias. Okay, I got more, but to begin with, obviously the Cotman Lab as a whole has been so fun. And obviously my committee members, you guys have come to all of my meetings, even the external. And I've gotten a lot of guidance on how to do my work better, how to think better. It's been really impactful. And then obviously the Varium staff, they're so good with the mice, and Harry's fabulous in terms of training. I'd never worked with animals before. I was an E. coli kid before this. And I love the mice. And in part, I think I get that from seeing Harry work with them. Here's the funding. Here's my favorite mouse, AF7. She's wonderful. And then more photos to the Cotman Lab, to Andreas. Thank you so much for welcoming me into the lab. I love this project. I'm really proud of the work that we've done. And I'm very excited for everyone to get to see what I've been doing for so long. There's been several people that have come through. I'll start with Dustin. In large part, I joined the lab because of you. You were so nice to me when I was rotating, and you were such a good mentor early on, and particularly with the fibrophotometry, you made it possible for us. So I want to make sure that your praises are sung. Santi, obviously, you make me think better about science, I think. I'm more of like a molecular in my training, and I think that in large part you've made me more of a neuroscientist. So I want to thank you. To the undergrad, or not undergrad, sorry, master's student, sorry. Sonia, who's on Zoom, she did so much training with me, and I really appreciated it. And she was a fantastic lab manager. The lab has never been the same since she left, because everything was color coordinated and organized. To Sasha, I'm so proud of you. So she is now doing her PhD at Rosalind Franklin University, and I get to get updates about what she's doing, and I'm so proud. To Kendra, you just graduated, and I'm so proud of just watching you grow as a scientist has been an honor. And then to all the undergrads I've got to work with, it's been really fun because thinking about myself as an undergrad and now I'm on the opposite side. It's very exciting. Obviously Alicia, I have a twin. She's in the back for some reason, but she moved here to New York City with me without ever being to New York City. And in many ways, because you've been here through all of this with me, it feels like this is our achievement. I'm like the communist twin of like, it's all of this. Yeah, and she's helped with some of the projects, and she's seen a lot of presentations, so. To my family, sorry. My mom and my aunt are here, my dad's on Zoom. I'm really lucky in that I have a lot of support with the family. I know that they're proud of me, and it means a lot. Also, I had a gerbil, but he has passed, but I think he's proud of me. To my friends, many of them are here. It's really made New York City feel like home, even though I'm definitely a West Coast kid still. I like recycling a little more than you guys. But yeah, I just really appreciate everyone. And some of these are also people that started as lab members and have now become friends. All the Christmas parties. And then also, as Andreas mentioned, I am part of Brainy and CUNY SciComm, and there's been so much joy that that's brought me. MBL, so last summer I was able to go to the neurobiology program. They sent me out for free, which I think was like $14,000 on their part. And that's the point, I think, where I became a neuroscientist. It was so impactful, and I'm so grateful for everything I learned there. And then miscellaneous, everyone else who supported me. My old lab from UW, I think is on Zoom right now. And it was the first place where I was like, maybe I can be a scientist, because I know I look a little goofy, but they always took me so seriously and they were so supportive. Alva, I did that as an undergrad, and I would not be where I am today without them. And then Moving Brains Foundation, where they teach Parkinson's patients how to box, led by Coach Reeves. It's wonderful. It's wonderful work, and it's really good to get out of the lab and then remember why you're doing what you're doing, because hanging out with the mice all day is fun, but there's actually applications and reasons it should be funded. And then last but not least, also these are videos. Please play. I have so many mouse pictures, I had to take the bed. I love the mice, everyone knows that. I'm a rodent person at heart, and I say it, but it's an honor to get to know all of them because they have their own personalities, and honestly, their work would not be possible without them. And that's it.
Excellent, thank you. Yeah, are there questions for Aria?
Alicia, question. Oh, God. Come on.
Yes.
I can make like super zoomed out, maybe. So how does the role of the ACE2 with COVID, like how do your results influence the way we perceive long COVID or the impact of ACE2?
Yeah, for sure. So the main thinking I have right now is that, and I didn't address this in the talk, it was pretty long, sorry. When you get COVID, there's an increase in ACE2 shedding. So as I showed in my talk, ACE2 is normally membrane bound. And so that's how you can have these like local expression, which I, through my work, shown is important. When you get COVID, it becomes loose from the membrane and becomes free floating. And so you have this loss of localized ACE2 now when you are currently with COVID or when you've recovered. And it takes a while to recover. And there's actually interesting work showing that if you look at people who had severe COVID or severe long COVID, they have more soluble ACE2 in their, both in the blood and the spinal fluid, as compared to someone who had not as bad of a case or control. So that's obviously circumstantial, but what my thinking is that the local ACE2 is particularly important, and that is what the virus is changing.
Thanks.
More questions? Yeah.
So you opened with this reminder that the pathways we've been thought of in terms of like blood pressure and stuff.
Mhm.
So with all this work, how in your mind have you come to think of how biology uses this pathway more generally, and how do you think about that?
Yeah, actually through writing my thesis, I thought a lot more about this because, as I said, like nature's cheap and a lot of stuff is reused. And this is a very ancient pathway. It's all the way back in like some sponges and stuff. And so I think there are these two different forms of it, and that the brain is doing its own thing, but it's just being reused. So I don't know evolutionarily what the main function was, but that's kind of how I think about it. And obviously, when you start thinking about treatment, you need to think about, you don't want to mess with people's blood pressure. So is there ways to particularly target one function of RAS versus the blood pressure side? I don't know if that answers that.
Any other questions? If not, then we thank everyone and please come back around 2.